I think though all our techniques are very different, you have people who you can talk to and get their advice on few things that you're going to use.
Yeah, especially the collaborative atmosphere of Mareusz labs and the campus, as well as the fantastic facilities, the great discussions. I think that really helps to understand your biological question.
Hi, my name is Aswini Panda, and I'm a PhD student in the Falk lab here at the Mareusz labs.
Hi, I'm María. I'm a PhD student in the lab of Krista Bucher.
And my name is Mateusz. I am a PhD student in the Quercus lab at the Mareusz, and all of us have recently been awarded the Doc Fellowship from the Austrian Academy of Sciences. So I think our PhD projects are quite different, we discussed, but there is certain aims that we think we have in common, and that is that we all want to understand how very basic biological processes make cells work as they should work under native conditions.
So genes need to be expressed at the correct time in the correct place in order to ensure the correct levels of gene expression for the cell to function properly and for development. And these regulatory elements, such as enhancers and promoters, are key players in this regulation. I'm studying how different enhancers and also their distance to the promoter play a role in the regulation of achieving the correct amount of gene expression level.
Of course, some genes have to be enhanced, the expression, and some genes have to be repressed, and this is the case very often with transposons. In the Quercus lab, we actually want to harness the power of transposons and their molecular activities in order to maybe make them in the future into next generation genome editing tools.
What I am interested in is to understand how this protein ERH, which is evolutionary conserved, and it's one of the highly expressed proteins in humans, how this regulates so many of this interaction with different proteins that are part of various biological pathways. I want to understand, using structural biology, how it mediates all this interaction with different proteins and regulates their function.
And for my project, I'm using molecular biology techniques and also synthetic biology in mass embryonic stem cells and a lot of microscopy.
In my project, I mostly use biochemistry as well as synthetic biology and structural biology, in particular single particle cryo-EM, to understand how the transposases work on a molecular level.